Greetings and welcome to Bozeman, Montana. I'm here coaching the ski team at Montana's Invitational, and as you can see, it is a winter wonderland. As you look around at all of this beautiful falling snow, it is a marvelous day to celebrate microbiology because, as it turns out, even snow is filled with microorganisms, and you may not have known it, but in fact, there are microorganisms out there that play a huge role in nucleating, making the core particle for snow and for ice. One such microorganism is called Pseudomonas syringae. Pseudomonas syringae forms what's called INA, or ice nucleation protein. This protein has one domain that serves to mimic the hexagonal structure of ice and to trigger the nucleation of that ice. In fact, I'm going to show you a sweet YouTube video that actually shows Pseudomonas syringae taking supercooled ice or supercooled water and converting it into ice. Because of that INA protein or ice nucleation protein, that protein is going to trigger this very chilled water to in fact become ice. Boom! Check it out! Isn't that the sweetest thing ever? So in fact, Pseudomonas syringae has all kinds of applications in ski areas where in many cases they use chemicals to seed the clouds. But in fact, Pseudomonas syringae could also be used to seed clouds. So when we think about snow, we have to now start to think about it as housing hundreds of even thousands of microorganisms. I've gone ahead and collected a sample of the Montana snow, and if we were to take this snow, we could isolate from it different microbial life. And let's talk for a minute how if we wanted to isolate just Pseudomonas syringae from this snow, how would we go about doing that? We're going to have the rest of that conversation from inside where it's a little bit warmer. Not only do samples like this one taken from snow harbor hundreds or even thousands of microorganisms, but also when we take a clinical sample, for example, if we were to take a throat swab or a urine sample, there would be hundreds and even thousands of microorganisms in those samples as well. Thus, it's really important for us to be able to separate those microorganisms that might be pathogenic from those that are non-pathogenic, to be able to differentiate, and then to be able to diagnose and perhaps to offer therapeutic strategies for controlling whatever it is that might be causing the infection. So we can see that the importance of isolating a pure culture is paramount, and that is going to be step number one in many of our procedures and in clinical microbiology labs. The first step is always to get a pure culture. Now, there are a number of ways in which we can do that, one of which you have already done. In fact, you'll recall doing the spread plate last time, where we did a series of dilutions of our E. coli culture in the sterile water bottles until we got to a low enough concentration that we could dip it onto the plate and we could spread it around and get isolation. You'll recall doing that last time, and you can see that that's what's going on in the background of this video. Of course, the epically fun dipping in the ethanol and flaming it out, and then spreading on the surface of the agar plate. So spread plates are absolutely one way in which we can get an isolated pure culture. However, they're certainly not the only way, and they are definitely not the easiest way. Let's talk about some other methods. First one is called the pour plate. Now, the pour plate is similar to a spread plate, except for in a pour plate, you actually put the bacteria that you've diluted into molten agar. So that is, the agar hasn't hardened yet, and the plate hasn't been poured yet. This is very similar to a spread plate, except for that, you might guess, the colonies actually develop inside of the agar, and in many cases they are subsurface in the agar. We're going to try this out later. But first, the most important method for developing a pure culture is the streak plate. Yes, you guessed it. Today we get to go streaking, and we are going to be carefully assessing your streaking technique. So you're going to want to rock this out, and you're literally going to be manually diluting your culture onto a plate. Now, be certain to find your way to virtual edge, and be absolutely sure to find your way to the experiment that shows the streaking demonstration, which is experiment number 13, and I want you to get really good with this streak plate. We are going to practice all semester until your streaking technique is absolutely perfect. So we want to have epic streakers. So what you're going to do with this technique is you're going to make a T on the bottom of your plate, and you're going to literally fill in the highly concentrated area in the top of the T. And then you're going to be pulling it down a gradient into these second and third areas. So you're actually literally manually diluting it across the surface of the plate. How easy is that? I mean, not only is streaking fun, but it's also soups easy. You're just going to sterilize your loop, and you're going to dip it in the culture, or you're going to take a colony from another plate, and you're just going to put it heavily in the top of the T. And then you're going to bring it down gradually throughout the rest of the regions. So that's basically going to manually dilute the culture. So the bacteria grow the closest together up in the top of the T, and then as we spread them through the second and third areas in the T, they get farther and farther apart from one another. That is, less and less of them fall off the loop all the time. There are fewer and fewer holding onto the loop every time you pass across the agar, until finally you get to the point that only a few are still holding onto the loop and a few are falling off, and then single cells land on the agar, and that single cell will grow into a colony. So it's pretty cool manual way. Streaking is the raddest way to separate bacteria and get pure cultures or single isolated colonies. It's cool to think about the fact that even though we may have streaked from, say, a throat swab or something like that, and we isolate them using a T-streak, it might not be that we get a completely pure culture the first time we T-streak. That is, we may want to streak again a second time. We may want to re-streak. So we're going to, the second lab, we're going to go ahead, or next lab, right, we're going to go ahead and pick up what looks like an isolated culture off of our T-streak, and we're going to re-streak it. It's considered good microbiological practice to always streak a second time, right? I mean, who would only want to go streaking once, right? You've got to go ahead and do it a second time. So pick up an isolated colony and re-streak it, because it's possible to have a minute level of contamination in your isolated colony. What if you've got one big bacterial colony and one tiny one, and the big one kind of obscures the tiny one, and it's hard to see the tiny one, so you might actually have more than one microorganism in what looks like a single colony. And sometimes we can tell a contaminated colony from another type of colony. Maybe it's got kind of an irregular shape, or maybe there's a little bit of a different color or different consistency. So it's just always a good idea to streak, not just once, but to streak a second time, streak and re-streak for good practice. So our look at microorganisms in the snow, which is starting to melt now, reminds us that different microorganisms have different temperature ranges over which they can thrive. In fact, there's a roughly 15 to 30 degree range over which microorganisms can grow. This particular graph shows that growth curve, and you'll notice that within that 15 to 30 degree range, there are three cardinal temperatures: the minimum, the optimum, and the maximum. Of course, we can guess that the optimum is the temperature at which the microorganism's growth rate is the highest, the minimum, the lowest temperature at which it can grow, and the maximum, the highest. Now, this growth curve represents the enzymatic activities of the cell. So you can see that as we rise from the minimum to the optimum, that's representing increased enzymatic velocity, and of course that is reflected in overall increased metabolic rate and increased growth rate. Now, when we take a look at the cardinal temperatures, it's worth noting that the optimum is always closer to the maximum than it is to the minimum. It's also very worth noting that the damage that is done to a cell below the minimum is very different than the kind of damage that's done to it above the maximum. That is, damage below the minimum is reversible. So the damage done below the minimum there are things like membrane solidification and malfunction of membrane proteins. However, the cells survive, and in fact, we take advantage of that feature of bacterial cells in that we store our bacteria in either liquid nitrogen or in a minus 80 freezer. We put a cryoprotectant in there and we put it in a cryo vial, and we store our organisms in sort of suspended animation below their minimum temperature. We can then take them out of those cryovials and streak them onto a medium of their choice, and they will grow again. That is absolutely not what happens above the maximum. Above the maximum temperature, irreversible denaturation of enzymes occurs, lipid bilayer disintegration, and once again, we take advantage of this as well in order to sterilize the media that we work with, all of the glassware and the tips and whatnot that you work with in lab. We use the autoclave, which raises the temperature to a very high temperature, 121 degrees Celsius, very high pressure, and we do irreversible damage, killing everything that might be living on whatever it is that we want to sterilize. So very different things that happen below the minimum versus above the maximum. Now, depending upon what kind of organism we're looking at, the 15 to 30 degree range over which we find these cardinal temperatures is going to be very, very different. So within the snow that we were looking at here in Bozeman, those microorganisms are going to thrive at temperatures with an optimum of maybe 15 degrees Celsius, but growing down to below zero. In fact, today at the race venue, the temperature of the snow was around -5 degrees Celsius. So that's really quite cold, but we know there's still bacteria thriving and growing in that temperature of snow. Perhaps it's not their optimum, but certainly it is not necessarily their minimum either. Now, on the flip side, something that lives and grows on our body is going to have an optimum temperature of roughly 37 degrees Celsius. That's body temperature, so that's going to be where its optimum temperature hits. Let's look at some of the classifications based upon temperature, and let's actually begin low. Let's start with what some of my TAs joke are the psychos out there, the psychrophiles, those that really love the low temperatures. So our snow microorganisms like temperatures of less than 0 degrees Celsius all the way to around 20 degrees Celsius with an optimum of maybe around 15 degrees Celsius. Now, the psychrophilic example that I'm giving here and showing a picture of is what we term bleeding footprints or watermelon snow. And this actually was a picture that I took at the base of Mount Massive near a lake called Rainbow Lake. And this picture was taken in August. So this was a long-lasting snowfield, one that lasted throughout the entire summer. And when you see the snow melt, a green type of algae, we actually see that it gets more and more concentrated, and you can see the algae, the psychrophilic algae, better and better. This also occurs up in the snowies, and in fact, this is a picture of me skiing, and you can see actually the band of the red colored snow. So the watermelon snow is really evident up in the snowies in the summer. This was at the end of June when we were skiing up in the snowies. So psychrophiles like it cold, and we can say also that psychrotrophs are like it to be pretty cold. So you can see there the next ones up. They like temperatures that are at an optimum at around 22 to 25, which this is room temperature. So room temperature sits right around there. They like growing the best at that temperature, but if you put them in the refrigerator, they keep on growing. Notice that they'll go down to 0 degrees Celsius as well, so their range encompasses colder temperatures. These are often our food spoilage microorganisms. So if you think about something like Listeria monocytogenes, which caused the epic cantaloupe scare in the Rocky Ford cantaloupes in 2011, maybe some of you remember that, is a psychotroph. It's a food spoilage, but also a pathogenic organism. Now we can talk about those bacteria that are mesophiles, which means that they like 37 degrees Celsius, our body temperature. And these are going to be more of our normal flora, our microbiota, but also our pathogens would fit into this category. not all of them, but many of them really prefer that 37 degrees Celsius temperature. Most of them are at least mesophilic, liking something maybe closer to 33 if it's something like Mycobacterium leprae that we talked about earlier that likes to grow on your fingers. They're a little bit cooler, but they're mesophilic. Now, at the very top end of the temperature scale, we have our thermophiles. The picture that you see here on the topmost upper right is a picture that I took not very far from Bozeman. If you head into Yellowstone National Park, you're going to see a lot of these epic thermophiles. And in Lower Geyser Basin, where this picture was taken, you're seeing a characteristic V pattern of growth where things like Thermus aquaticus, a very heat-loving bacterium, will grow in the white region in the V, whereas the less heat-tolerant organisms will grow on the edge of the V, and these are more, they're photosynthetic, and you can see they're forming a lot of accessory pigments in this particular picture. So these are thermophiles liking it hot, but not quite as hot as hyperthermophiles. So if we go deep down into a hydrothermal vent where the pressure is such that the temperature of the water can exceed boiling, then we start to see those organisms that like and enjoy temperatures of 115 degrees Celsius or even hotter. Oftentimes their optimum is 85, but they can be put into an autoclave. We could put them in an environment that would kill everything else, and the hyperthermophile would keep on living. So we understand that temperature is one of the major environmental factors that affects microbial growth, but by no means is it the only one. And of course, you can think about the idea that pH, or hydrogen ion concentration, especially when we think about the bacteria and algae of Yellowstone National Park, we see that there are a lot of organisms in those environments that like acidic pHs, high hydrogen ion concentrations. And in fact, this is a picture that I took also in Yellowstone, a different region than the other picture. This is in Lemonade Springs, which is near Norris Geyser Basin, if you happen to have been there. And you can see that this is a very, very acid lover because cyanidium will tolerate pHs of 0. Check that. That's crazy. Really, really low pHs that we would not survive. Most of the bacteria that live on us are more neutrophilic, though we do have some bacteria in our gut that tolerate very, very low pHs. And then we do have some body regions that are even more basic. That is higher pH, the lower hydrogen ion concentration. Alkalophiles, spelled two different ways depending upon the textbook you consult, alkalophiles or alkaliphiles, but in all means the same thing, liking higher pH values. Now, you all are going to be doing a little bit of mystery solving. We've given you two bacteria, one that was isolated from this mine drainage that you see on the left, and actually this is right outside of my hometown of Leadville. You can actually see the mountains in the background, Mount Massive, Mount Albert. This is a mine drainage that is on the bike path where we like to roller ski in the summer. So we were at a ski training camp, we took our gloves and our sample vials, we hopped over the fence and we grabbed a sample from this mine drainage. And then over on the right is a picture of a lake bed. You will recognize this as just being just outside of Laramie on the way. If you happen to be biking out to Centennial, you'll find that. And we isolated a bacterium from this lake bed. And so you are going to investigate which of these is an acidophile and which is an alkalophile. And you'll come back with some information when you get your lab results. So hydrogen ion concentration, temperature, these are environmental factors that affect microbial growth, but so too is water availability. Have you ever thought about why it is that you can leave peanut butter on the shelf for long periods of time? Of course, some of it certainly has a lot of preservatives in it. But the reason that peanut butter, even without preservatives, doesn't go bad very rapidly is because there is very little water available. The same is true of salty solutions, and that's where we want to turn our attention, is to The osmotic pressure, the pressure that water exerts on the plasma membrane of an organism, and it's in response to the unequal distribution of dissolved solutes. So perhaps some of you are fans of poetry, and you know Samuel J. Coleridge's famous poem, "Water, water everywhere, but not a drop to drink. Water, water everywhere, and how the boards did shrink." So that actually is a poem that is all about osmotic pressure. And in that poem, the water is rushing out of the board cells, and it's causing them to shrink because the water is following the solutes and it's leaving the cell. So water always follows the higher concentration solute and moves towards the higher concentration solute, and that movement of water is called osmosis. So let's actually begin by looking at the hypertonic environment that the poem is all about, and then we'll come back and look at the hypotonic environment. So we're skipping ahead in your notes and looking at this environment where the boards did shrink. So seeing here that if you're in an environment that's very salty, like the seawater, the water rushes out from the cell. And so it's like we can't drink ocean water, right? Because it's so salty. And it's not because the ocean's not full of water. There's so much water in the ocean, but we can't drink it because all of that water is tied up in solute interactions. The water is unavailable to us. Hypertonic environments cause the boards to shrink, cause water to rush out of the cell, and cause the cytoplasmic membrane to dehydrate, to pull away from the cell wall, to shrink, and this is called plasmolysis. So there's that term that came straight from the poetry, shrinkage. So dehydrate, shrinks, pulls away from the cell wall. So this is generally the condition that does completely kill cells, and this is something you know very well. Those of you who are hunters and you make beef jerky or elk jerky and or antelope jerky, and you know that if you put salt onto that meat, it preserves the meat. We don't have to refrigerate anymore. And that's because the plasmolysis is killing bacterial contaminants. It's also preserving the meat, or it's preserving the meat by keeping the bacteria from thriving, but it's also drying the meat as well, and making the jerky kind of consistency. So that has everything to do with water availability. So in a hypertonic environment, water is unavailable. It's present, but it's not available because of the solute interactions. Organisms that can tolerate salty environments are called osmotolerant or halotolerant specifically. Osmotolerant, it refers to any kind of solute, whereas halotolerance specifically refers to salt. So something that can tolerate high concentrations of solute, we say it's osmotolerant. Something that can tolerate high concentrations of salt, we say is halotolerant. Now, there are some organisms that are absolutely crazy that can actually require salt. These are halophiles, salt lovers, and many of them have to have 25% sodium chloride in order to live. And in fact, we're going to be working with one of those, Halobacterium salinarium. So you're going to get the chance to actually see a halophilic archaean that you'll be working with on your plates. So let's look at the opposite, which actually bacteria quite enjoy a hypotonic environment. So an environment that has lower solute concentrations is called hypotonic. So let's take a look at that environment. In that environment, of course, water rushes in. Now that would burst a cell if the cell didn't have a cell wall. But remember that most bacteria enjoy a cell wall, the benefits of the cell wall and the rigid peptidoglycan found in that cell wall. So the hypotonic environment just causes the membrane to push up against the cell wall and to provide a certain amount of turgor pressure. Bacteria actually quite enjoy that turgor pressure, and in fact it ensures water. So this is actually not as deleterious. Now, if they lacked a cell wall, or if the cell wall lacked integrity, or if for some reason this just kept on pushing and it continued ad nauseum, then the cell might burst. But of course, as we mentioned, most bacteria, algae, and fungi also tolerate this and enjoy it. They synthesize solutes maybe that will cause this to occur so that they can maintain that turgor pressure. So it's all about the uneven distribution of solutes across a cell wall. It's of note that those bacteria that are osmotolerant are capable of synthesizing more solutes within the cell to allow them to bring in water even in an environment that's very hypertonic. That's actually how they enable life within that kind of high solute environment. We have one last environmental influence to talk about, and so we'll go ahead and move in that direction. And that's not to say this is the last of all of the things in the environment that might affect a cell, but we're going to look at oxygen concentration. And when we say oxygen, we mean diatomic O2 gas, because remember, it is naturally converted to toxic hydrogen peroxide and superoxide. And therefore, oxygen, although we think about it as life-giving, it's our terminal electron acceptor, oxygen is actually toxic. And those bacteria that cannot detoxify the toxic forms of oxygen, they die in the presence of that oxygen. So we have some bacteria that tolerate it and have to have it, some that tolerate it, some that have to have it, some that have to have a little bit, and some that can't really have any or they'll die. So our aerobes, our obligate aerobes, are those that require oxygen for growth. For example, like us, they may use it as a terminal electron acceptor, and so they have to have it. Something like Bacillus subtilis, something like Pseudomonas, these are good examples of organisms that live aerobically. They live in the top regions of a gradient when placed into a gradient. And one of the best ways to do this is actually to make a solution of semi-solid agar. And we usually use something called brain and heart infusion, which is what BHI stands for. And so it's an infusion, it's an extract from brain and heart. And so we put that into a gradient where the oxygen is present in high concentrations at the top, and then very low concentrations in the middle, and then finally no oxygen in the bottom. And so we can see that if the growth is up top, that's aerobic. If an organism grows throughout the tube, but more thickly on the top, then those are facultative anaerobes. They don't require it, but they like it. They really like to have it. And remember that oxygen is the best terminal electron acceptor. So even if an organism like E. coli can use something other than oxygen as its terminal electron acceptor, it prefers oxygen. You could say oxygen is the most badass terminal electron acceptor. It has a higher standard reduction potential. It makes the best acceptor of electrons. So in a facultative anaerobe, they like to use oxygen, but if they're pressured, they can use something else. And so they'll grow throughout. So facultative anaerobes will go throughout, but heavier on the top. Microaerophiles include things like Campylobacter jejuni, which is the bacterium that causes a lot of cases of enteric disease, very severe diarrhea. My sister suffered from Campylobacter jejuni. She had such watery diarrhea that I was trying to keep her hydrated with Gatorade, and she would drink blue Gatorade, and it would go in looking like blue Gatorade, and it would come right back out looking like blue Gatorade. And I apologize if this is the second time you're hearing that story, but it was pretty crazy, and that's a microaerophilic bacterium. So you can see how it would find niches in the gut that would have 2 to 10% oxygen, low concentrations of oxygen in which these organisms thrive. So they form this like thin band of growth in the region where they find their 2 to 10% oxygen, but they won't grow anywhere else. Now, anaerobes, of course, will only grow in the bottom of the tube, where we'll see that they're away from, or they're the ones that are killed by the toxic derivatives of oxygen. Now, aerotolerant anaerobes are indifferent to oxygen. They don't really care whether it's present or not, so their growth is unaffected by the oxygen gradient. Now, that includes things like our lactic acid bacteria, for example, things like lactobacillus and lactococcus, and the things that you see on your probiotic foods that you eat. These are aerotolerant. They don't care whether it is present or not. They're indifferent to it. That's because they only use fermentation and they don't respire at all. So that's their key to not caring about oxygen in their environment. So that is going to be the end of our conversation of environmental factors that affect bacterial growth.